Welcome. This video is going to look at 15.2 Part B on penicillin. The first part looked at aspirin, but penicillin is an antibiotic, which is a whole new class of medicines that were developed in the early 1900s. In the late 1800s, scientists realized that certain chemicals could kill pathogens, and it was the discovery of penicillin that led to the development of this whole class of antibiotics and really revolutionized medicine. Antibiotics are chemicals usually produced by microorganisms which act against other microorganisms, in this case bacteria. And discovery is credited to Fleming, who in 1928 was working with bacterial cultures and noticed mold had contaminated some work, but he also realized it stopped the bacteria from growing. So Fleming did not investigate what or how the antibacterial was working, but he did note that there was an antibacterial produced by this mold. So in the 1930s, Flory and Chain began working with the penicillin, injected into live mice, and found it cured bacterial infections. And they were able to isolate the penicillin from the mold that produced it, and they were able to produce it themselves in the lab, but they still didn't understand how it worked. In 1941, though, they went ahead and used it on the first humans, partly because of the huge need that was present due to World War II going on. In fact, the need was so great and penicillin was so scarce that it was actually recovered and reused from the urine of patients receiving penicillin, because up to 80% of the penicillin passed through their bodies without being metabolized. So in 1945, Dorothy Hodgkin determined the structure of penicillin G, which was Fleming's original penicillin. And the R group can be lots of different things. It's just like in organic chemistry, that can be anything. But the important thing is that the functional groups include, you've got an amine group here, an NH attached to, it's a secondary amine attached to two different carbon groups. And then you've got an amide group here. And then you've also got two methyl groups and a carboxyl group. And you may or may not notice there's a sulfur in there. The R group, the nice thing is that if you have different R groups, it gives us different kinds of penicillins like amoxicillin or Z-Pax, etc. But then the really unusual thing is this square structure here. It's called a ring, but it is indeed square. And it's the part that makes penicillin so unique and effective. It's got 3 carbon and 1 nitrogen, but the unique thing is that they're at 90-degree bond angles. So it's known as cyclic amide, or more often beta-lactam. And the 90-degree bond angle is unusual because typically these atoms form 120-degree or 109.5-degree trigonal or tetrahedral bonds. So this 90-degree bond angle makes these bonds very unstable or reactive. Bacteria needs an enzyme, transpeptidase, to form strong cell walls, but the beta-lactam ring reacts and binds this enzyme when it's near it, and this means that the bacteria can't make a strong cell wall, and the cells end up rupturing and dying. So if we look at the structures down here, you see that what happens is the enzyme binds on here, gets trapped, and deactivated. This ring opens up in the process of doing that, which is fine. That was its job. But then the cell wall, the bacteria cell wall, is going to be much weakened, and it's going to end up in killing the cell or rupturing it. Penicillin is effective for a wide range of bacteria. Infections of the ears, nose, and throat, mouth, wounds. But the big disadvantage is it's broken down by stomach acid. So to use penicillin, it has to be injected directly into the blood. But remember that R group, if you modify the side chain, then you can develop other antibiotics that we can take orally, like amoxicillin. Development of antibiotics has also led to greater antibiotic resistance. As early as the 1940s, it was known that some bacteria are resistant to penicillin or not killed off by it. And we now know that the same mechanism, the reactive beta-lactam ring, that makes penicillin so good at killing off bacteria is also what allows bacteria to resist it. Resistant bacteria produce an enzyme called penicillinase, which can open the beta-lactam ring before the penicillin does it to the, to the bacteria's enzyme. So it kind of turns the table on the penicillin. It opens up the ring, so now the penicillin cannot bind to the enzyme and render the bacteria with a weak cell wall. Now, this antibiotic resistance should only arise through genetic mutation and be in a small portion of the bacteria. But instead, we've seen a development of these superbugs, these highly resistant bacteria, just explode over. We now know that there's not just one enzyme like penicillinase that renders penicillin ineffective, but there's several different enzymes that these resistant bacteria can have. So we call these enzymes beta-lactamases because they render the beta-lactamase ineffective. And the total number of different beta-lactamases and the total number of these superbugs has skyrocketed. You can see that on the chart below that in 1990 it really started taking off. And scientists feel it's due to a number of factors. Antibiotics being overprescribed, people not finishing all their antibiotic, using antibiotics in animal feeds, and using antibacterial soaps and different cleansers in our home. All the above kill the weak bacteria while allowing the stronger resistant bacteria to reproduce and thrive. So it's really the survival of the fittest in this case. So what are some solutions or responses to the resistant bacteria? We can continue to develop different forms of penicillin that are resistant to the beta-lactamases. We can control and restrict the use of antibiotics, making them prescription only and encouraging doctors not to overprescribe their use. And we can educate patients on the importance of completing the full course of an antibacterial or of an antibiotic treatment. Okay, because when you don't complete it, when you quit taking it as you feel better, now the strongest bacteria are the ones that didn't get killed off, and they're the ones that are going to go on to reproduce.